Hey everyone, welcome back to the GEP Pathways video series. Today we will be going over part 6.4, which reviews how to use spliced RNA-seq reads to verify the coordinates for intron 1. This figure here gives us a general overview of RNA-seq reads. Since RNA-seq reads are derived primarily from processed mRNAs where the introns have been removed, the subset of RNA-seq reads that span multiple exons can provide us with additional evidence for a splice junction. When we map a spliced RNA-seq read against the scaffold, part of the spliced read will map to one CDS while the rest of the read will map to another. The region between these two alignment blocks corresponds to the intron. Part A shows us the processed mRNA that has had its intron spliced out. Part B illustrates the processed mRNA fragmented into smaller pieces called reads, which are then sequenced, meaning the order of the bases gets determined. Part C depicts the mapped reads against a genomic scaffold looking for overlaps in the coding sequences. And Part D illustrates how a computer program might analyze the mapping data and infer the possible locations of the splice junctions. So let's go back to our genome browser for REB. If we scroll down under the RNA-seq tracks, let's change the combined splice junctions track to PAC in order for us to utilize the RNA-seq to verify the coordinates for this intron. Note that the combined splice junctions track shows splice junctions extracted from spliced RNA-seq reads that have been aligned to the genome. Now let's click on the refresh button to apply the changes. To examine the region surrounding intron 1, enter NC underscore 052530 colon 19150858 dash 19150984 into the search terms text box and click Go. Remember that we found these coordinates in part 6.3. Since our analysis in part 6.3 suggested that CDS1 ends at 19150857, intron 1 should begin at 19150858. Furthermore, we found that CDS2 begins at 19150985, thus intron 1 should end at 19150984. Now, let's zoom out 3 times to examine the 381 base pairs surrounding this position. Here we can see that there is only one splice junction predicted in this region as shown by the red arrowed line between the CDSs. The predicted splice donor and acceptor sites are indicated by the purple and orange arrows respectively. To examine the splice donor site predicted by the splice junction 00113127, zoom into the region surrounding the beginning of the intron predicted by this junction. Here we can see that in reading frame +3, we have a phase 1 GT donor. Notice that all of our tracks support the location of this donor site. To examine the splice acceptor site predicted by the splice junction, zoom into the region surrounding the end of intron 1. Since we are in reading frame +1, we can see that our AG acceptor phase is 2. Notice again that all of our tracks support the location of this acceptor site. So this splice junction between the phase 1 donor site of CDS1 and the phase 2 acceptor site of CDS2 is supported by the NCBI RefSeq genes and spawn alignment of D-melanogaster proteins, gene predictors Gemoma, NSCAN, and Augustus, as well as our RNA-seq data. We can gather additional evidence to support this splice junction by clicking on JUNC 00113127, and then we examine the score field. The score tells us how many spliced RNA-seq reads there are that support a predicted splice junction. Since this junction has a score of 6,257, that splice junction prediction is supported by 6,257 spliced RNA-seq reads. Splice junction predictions are color-coded based on the number of spliced RNA-seq reads that support the junction. Based on the color-coded table in the description section, our junction will be red in the Genome Browser image since greater than 1,000 spliced RNA-seq reads support the feature. The higher the read, the better. Based on multiple lines of evidence, we can conclude the splice junction prediction is consistent with our splice donor site for CDS1 at 19,150,858 to 19,150,859, and our splice acceptor site for CDS2 at 19,150,983 to 19,150,984 that we annotated in part 6.3.